Today, we're talking about something that sounds like it's straight out of a sci-fi movie. It's a revolutionary AI that's not just reading the code of life, but it's actually starting to write it. Yeah, this is the story of a model that can generate brand new biology, literally from scratch. Let's dive in.
And you know what? To really show you how wild this is, we're not going to save the best for last. We are starting with the most mind-blowing result right out of the gate.
So let's just ask the big question right up front. Is it even possible for an AI to design a totally new, completely functional biological machine? Something that nature has never cooked up on its own. I mean, it sounds like pure fantasy, right?
Well, it's not. Meet EvoCas91. This is a real working CRISPR gene editing system. Think of it like a pair of microscopic programmable scissors for DNA. And here's the kicker. It was designed from the ground up by an artificial intelligence. They tested it in a lab, and it actually works. This is happening.
Okay, so now that you've seen what it can do, let's hit the brakes and back up a little bit. How on earth did we get here? To really get why this is such a huge deal, you gotta understand the problem this AI was built to solve.
So all the instructions for every living thing are written in DNA. You can think of it like a biological programming language, but it's one that is just unbelievably complex. I mean, even a simple genome has millions of these characters, these base pairs, and they all have to work together.
And that's where the old way of doing things really struggled. See, previous AIs tried to read DNA by sort of squinting at it. They would group letters into these chunks called tokens. It's like trying to read a novel by only looking at one word at a time. You see the words, sure, but you have no idea what the story is about. You completely miss the plot. But the new model, Evo, it's totally different. It reads every single letter with perfect precision, and it can see these huge stretches of code all at once. It's like going from reading one word at a time to reading entire chapters. It gets the whole story.
So with that, let's properly introduce the star of the show, the AI model they call Evo.
You know what? The easiest way to think about Evo is to compare it to something like ChatGPT. It's a large language model, but instead of training on Wikipedia and the rest of the internet, it was trained on the grammar of life itself, on thousands and thousands of real genomes.
And the scale we're talking about here is just staggering. Evo was trained on a data set of 300 billion nucleotides. That's 300 billion individual letters of DNA code pulled from tens of thousands of different organisms. It basically sat down and read a giant chunk of life's library.
And this right here, this is its superpower. In one single pass, Evo can read and process a sequence of over 131,000 letters of DNA. That massive context window is the key. It's what lets it see how a gene way over here might be talking to a gene way over there.
So how does it do that? Well, the secret sauce is a brand new AI architecture called striped hyena. Now, we don't need to get into the technical weeds here, but what you need to know is that it is ridiculously efficient. It's what makes it possible to analyze these enormous biological sequences at single letter detail, something that was just a computational nightmare before.
Okay, so Evo has this incredibly deep understanding of DNA's language, but what can you actually do with that? This is where it goes from just being cool to being a total game changer. It's so much more than just reading the code. Just look at this chart. It's showing how well different AIs can predict if a piece of DNA is going to act like an on switch for a gene. Now, the magic words here are zero shot. That means Evo did this cold. It had zero specific training for this task. And look at that first bar. It performed almost as well as the specialized models in the second bar that were custom built to do only this one thing. That's how well it just understands biology.
And that deep understanding is what allows it to become a creator. So let's bring it all back to that big reveal from the beginning. Using its knowledge, Evo didn't just find a new CRISPR system, it generated the designs for completely new ones. These complex little machines made of protein and RNA that nobody had ever seen before. And this wasn't some lucky fluke. The researchers also challenged it to design another complex machine called a transposon. You can think of them as jumping genes that hop around the genome. They gave it a shot, and out of 24 designs, 11 of them actually worked in the lab. That's almost a 50% success rate for an unbelievably hard design problem.
This quote from the research paper, man, it really just says it all. We're not just limited to finding the tools that evolution happened to create anymore. We are now officially at the point where we can start designing our own biological tools, ones that go beyond anything found in nature.
So here's the thing. Evo doesn't just get the little details, it understands the whole system. It can actually predict which genes are absolutely critical for an organism to even survive, just by reading its DNA. And its predictions get way, way better when it can see the gene's neighborhood, the surrounding context. That proves it's not just looking at genes in isolation. It understands how they all fit together to make a living thing live.
So let's zoom out. What does this all actually mean for the future? Because this isn't just a cool science experiment. The implications here are, well, they're profound.
I mean, the potential here is just absolutely staggering. We're talking about a future where we could design new medicines for specific diseases, create custom enzymes that could literally eat plastic waste, or brew out biofuels. And maybe most importantly, we could unlock a fundamentally deeper understanding of the operating system of life itself. But of course, with a tool this powerful comes a huge responsibility. And the researchers who built Evo are being very clear about this. They're actively calling for scientists, for security experts, for policymakers to get together and have a serious conversation about biosafety and ethics now, to make sure this technology is developed in a safe and responsible way.
Which kind of leaves us with this final thought. For the very first time, we're shifting from being just readers of the genetic code to becoming writers. We're learning how to write the book of life. And the question for every single one of us is, what story should we tell?